Welcome back to the last video of Unit 1. This one is about the scientific method. The scientific method is a way that we use and scientists use to figure out answers to problems. And it all starts off with curiosity. We have to wonder about things so that we can know exactly what we're going to start learning about, what we're going to start experimenting on. Now, you already have, hopefully, in your lab book, you've got your topic at the top, you got your date at the top, and you have now room for your main ideas and concepts. Maybe you're going to write things down as I talk, or maybe you're just going to listen through, and then you will go back through the slide deck and write things down later. That is completely up to you. So we start off with a question. Scientific method, the scientific method begins with a question that you're going to be researching. We are going to be doing in class, or you at home are going to be doing the question that's at the top left. What affects reaction time? But there are many different questions that can be used in different experiments, some of which are listed here, others you can just come up with on your own. After you have a question, you need to gather some information. You might do some research if you don't know too much about the topic. Even if you do know about the topic, you want to do a little research and just make sure that what you know is actually true about the things that you're going to be learning about. Researching helps you to come up with an educated guess or hypothesis about what might happen. Also, as you're gathering information, you want to come up with what are your controls, what are your constants, what are your variables. The controls are things that you're comparing to. If you were doing a controlled experiment, you are comparing something to the way it's supposed to be. So for instance, if you're growing a plant and you're trying to see whether you can water it with Coke, you test watering it with Coca-Cola and you test watering it with water to see which one turns out better. And you compare your variable, watering it with a different substance, to the ones that are watered with water to see if it's better or worse. The other types of experiments are just comparing two things, like you're testing to see whether boys or girls, whether people who are less than 13 or more than 13, whether one thing is better than another. So you have constants and variables. Constants stay the same to keep it fair, and it's very important that you keep them the same to keep it fair. And variables change, and you can only have one variable. Now, going back to the experiment with plants, if you're watering plants with different substances, you can water the plant with water, orange juice, Coke. You can water it with milk. You can water it with Diet Dr. Pepper. Whatever you want to, you can use all those different liquids. But all you're doing is testing one thing, that variable of what liquid plants use best. Now, a little bit more about variables. I see I need to move myself out of the way of this little thing. There are independent variables and there are dependent variables. The independent variable is the variable that you are going to change. The dependent variable is the thing that changes as a result of the change you made in the experiment. So independent variables, some examples like gender. You can decide whether boys or girls are better. The amount of water, which I just talked about, or the type of liquid, the temperature, the location, the size, the shade, or the shape. The dependent variable are the things then that you're measuring based on those variables. So it might be like the speed of something, the amount of growth, the number of something. So if you look at the example right below, if I change the temperature, how will it affect plant growth? The independent variable is the temperature, and the dependent variable is the height of the plant. So hopefully that helps to determine the difference between dependent and independent variables. Now we move through a few of the vocab words. The control is something you compare to. So it's like the normal way of doing it. When you're in physical education class, you do some tests each year where you run the mile and you do the shuttle run. And basically you are comparing your time to other people who have been in 7th grade and are your age before you. That's what the control is. And they have different levels that you need to get to reach certain levels. We have constants. Those are things that are kept the same. It wouldn't be fair if I gave a test in science class and one group, while they were taking the test, would have complete quiet and be able to concentrate. And the other test, we would loose the monkeys. We'd let them run around the room and grab your paper and grab your pencil and shake the tables. That wouldn't be fair. So in a controlled experiment, you need to make sure that there are some things that are the same so that you don't make the test results different. The variable then is the condition that changes. So after you have all that, you're ready to make a hypothesis. A hypothesis is basically the answer to your question. So if your question is, do boys or girls have a faster reaction time? Your answer would simply state that boys have a faster reaction time, because we all, or we could say girls have a faster reaction time too, if you'd rather. Either way is fine, but just answer the question. You don't have to explain, you don't have to say anything else, but it should be a complete sentence that explains your answer to the question. A hypothesis is a prediction that can be tested. So we're making that hypothesis, making that guess so that we can test it and see if it will actually work out the way that we thought it would. Now we're ready to actually get ready for our experiment. We need to plan. We need to look at what experiment we're going to do to test that. We need to gather our materials. We need to kind of decide who or what things we're going to be testing. Those are all important things in the planning stage. Once you have it all planned, then we come to the part that everyone really likes. It's actually gathering data. It's doing the experiment and recording things and finding out what really happens. So that's where you're doing recording, you're organizing things in a lab book, on a sheet of paper, on a computer maybe, keeping track of the information that you gathered while you were doing your experiment. After you have your data, then you get to the part that, just to tell you, 7th graders aren't really good at. That's called the analyzing. That's where you actually make your charts and your graphs and you put things together and you look at things carefully and you try to figure out which of my variables actually turned out to be the best, because that then leads you towards your conclusion. And sometimes it means looking at what might have gone wrong in the experiment that might have changed the results. Sometimes it means throwing out different results that you got if one result seems to be completely different than all of the other ones. So analyzing is a really very important part of doing your experiment, because that way you come up with a conclusion that is actually an accurate conclusion. Another vocab word, because we're collecting data as we're experimenting. So we finally end up with a conclusion. And in a conclusion, there's basically 4 things that you need to talk about. Number 1, was your hypothesis correct? This would be where you could then also say, I thought that this was going to happen because of this and this and this, but I found out that that was wrong. Or I thought this was going to happen because of this and this and this, but it actually turned out that it was that way. And so I was right. You need to explain your data. You need to give numbers. You need to say, on average, the plants that were grown with water had a 3.4 centimeter height, or 3.4 centimeters in height, and the ones grown in milk had only about a 2.2 centimeters in height. And then you might even tell what the difference was between those numbers. So that's explaining your data. You do not need to go through and talk about each trial that you did, but you should at least explain the averages and the ones that are important in determining whether your hypothesis was correct or not. What are some of the sources of error? What might have gone wrong? What might have you done wrong? What might have the computer done wrong? There's lots of different possibilities. Did the bell ring while you were testing something? Did it start to rain? There are all kinds of sources of error that might have happened. Did a volcano explode? It could have happened while you were testing your project. And then what else could you do? What further study could you do on that topic? So if you're testing reaction time to see whether boys or girls have a faster reaction time, then you might move on and say, okay, is my left hand or is my right hand faster? Or do I react faster to something I hear? Or do I react faster to something I can see? Those are all things you might do in your further study of your conclusion. It should be in paragraph form, and please, it shouldn't just seem like you're answering 4 questions. It should be a flowing paragraph that explains about your experiment and the results that you got. Now it's your turn to write a few questions, like a little quiz about the scientific method. And when you get done with that, if you have any further questions that you would like clarification on, write those down at the bottom and maybe send me a message or put that in your daily check. And I can answer that for you. Maybe make another video, maybe just send you a message. Maybe I can send you to a YouTube video or somewhere else online that you can find the answers or help you to understand that topic a little bit better. So this concludes Unit 1. Be ready for the test coming up sometime soon. And then look for new videos about the cells unit after that.